My dear students, welcome to Arts College Studio. Today in this class, we are going to learn about nutrition in protozoa. What is nutrition? Nutrition is nothing but supplying food materials. It is comprising of so many phases like ingestion, digestion, absorption, and assimilation. Although protozoans do not have any specialized digestive system because of their unicellular nature, the digestion is completed within the cell itself. As a result, you can find a wonderful dimension of nutrition in protozoans. Now let us see what are the phases involved in the nutrition of the protozoans. Here you can see, I have already explained that protozoan nutrition comprises of some phases like ingestion, digestion, absorption, assimilation, ultimately egestion. Now, if you take the process of nutrition in protozoans, protozoans exhibit much diversity in this nutrition. Here you can see nutrition in protozoa exhibits much diversity. It may be holozoic, or it may be holophytic, or saprozoic, parasitic, or mixotropic. So like that, there are nearly 5 different types of nutrition in case of protozoans. One is holozoic, holophytic, saprozoic, parasitic, mixotropic. We will explain one by one. What is holozoic mode of nutrition? Holozoic mode of nutrition is animal mode of nutrition. That means as the animal consumes, as the animal devours the prey, protozoans also devour the prey. That is called holozoic mode of nutrition. It is heterotrophic. Coming to the holophytic, it is the plant mode of nutrition. That means they can prepare their own food material by using chlorophyll or some other pigment like that. They can trap the sunlight and they can perform photosynthesis and they can fix it up, convert the same into the chemical energy. To be very brief, holozoic mode of nutrition is animal mode of nutrition. Holophytic mode of nutrition is the plant mode of nutrition. And coming to saprozoic, it is also called osmotrophic mode of nutrition. Saprozoic mode of nutrition is the nutrition where the animal can absorb dead and decaying organic matter from the surface. Parasitic mode of nutrition means it consumes the food taken by the host. There are many protozoans which lead a parasitic life. For example, you can take Entamoeba histolytica or Plasmodium species like that. They are present inside the host and they can rob the food of the host. And there is mixotrophic mode of nutrition, which is very unique. Mixotrophic mode of nutrition is the nutrition that involves more than one type of nutrition. That means if any protozoan exhibits more than one type of nutrition, then it is called mixotrophic mode of nutrition. We will try to provide the example at appropriate time. Now, let us explain one by one. Here you can see holozoic mode of nutrition is animal mode of nutrition. That means it is not autotrophic, it is zootrophic and heterotrophic. That means it depends, it devours, it consumes other food particles. These food particles may be either living or non-living. Normally, there are certain food choices even for protozoans. That means they are very selective in eating something. That means their menu is totally different. Let us see what are the food particles that can be preferred by the protozoans. Now, here you can see solid food particles may be taken, or sometimes even liquid is also just sucked. Intake of solid food particles is known as phagocytosis, and just drinking of the liquid is called pinocytosis. Here you can see solid food particles are ingested and digested. There are various modes by which the protozoans can ingest and digest the food material. But here you can see the menu of the protozoans. That means diatoms, favorites of protozoans, rotifers, crustaceans. Crustaceans belong to arthropoda. Other protozoans. That means a protozoa can devour the other protozoan species. So these are the menu that are available at the dining table of the protozoans. So let me repeat, diatoms, rotifers, crustaceans, and other protozoans may serve as food particles for the protozoans. And now here you can see the different phases of nutrition of protozoans. One is food capture. This is very important. Food should be captured. Protozoans can capture mobile food in one manner, and for capturing the immobile food, they may follow different strategies. Ingestion. That means ingestion is nothing but taking the food into the organ of ingestion. There is no special organ of ingestion like mouth in protozoans, but food particle will simply be ingested by using pseudopodia or other capturing devices. Coming to digestion, because these protozoans are unicellular in nature, the digestion occurs within the cell, especially in a vacuole called food vacuole. Hence, the digestion is called intracellular digestion. And after digestion, the digested food will be assimilated and absorbed, of course. First it is absorbed, later it is assimilated. And undigested waste material, undigested food particles will be simply adjusted through either temporary cell LNS or permanent cell LNS. You will see it later. And let us see how the protozoans will capture the prey. Capturing and ingesting the solid food materials, food particles, is called phagocytosis. Phagocytosis is nothing but intake of solid food particles. It is also called phagotrophy. And now here you can see there are various methods of phagocytosis. One is circumvallation. In this method, prey is captured by forming a food cup by throwing pseudopodia around the prey. Around the prey, pseudopodia are simply thrown out. And it is used for capturing the mobile food. Circumvallation is a method by which protozoans can capture the mobile prey. Here, let me repeat, in this method, prey is captured by forming a food cup. That means a food cup is formed by throwing pseudopodia. How this food cup is formed? It is due to the throwing of the pseudopodia, the food cup is formed. And active prey is captured in this method called circumvallation. I'll show the diagram later. Coming to the circumfluence, this method is used to capture immobile prey. That is a basic difference between circumvallation and circumfluence. In circumvallation, active prey is captured, whereas in case of circumfluence, immobile prey is captured. So in circumvallation, the prey is captured by forming a food cup by simply throwing away the pseudopodia. And here there is no contact with the prey. That means the cytoplasm or pseudopodia do not come in contact with the prey. Just the pseudopodia are thrown around the prey and it's simply engulfed. And coming to the circumfluence, this method is used to capture immobile prey. In this method, the food cup, no doubt, here also it is formed. How it advances is, once it comes in contact with the prey, that means once the mobile or immobile prey, not mobile prey, once the immobile prey comes in contact with the cytoplasm of the protozoan. Now what will happen? Once it comes in contact with the prey, food cup is formed and it advances and the cytoplasm flows around to engulf it. Here you can see, here also food cup is formed and even in circumfluence food cup is formed, but here in the former one, in circumvallation, there is no contact between the prey and the food cup. Simply the pseudopodia are thrown and prey is engulfed. Here in case of circumfluence, once the protozoan comes in contact with the prey, the two immobile prey, food cup is formed and the prey is engulfed. That means it is simply ingested. So this is how these two methods will work out. And here you can find the diagrammatic representation of both circumvallation and circumfluence. Here you can see there is a food particle. This is a mobile food particle, movable food particle. Pseudopodia are being thrown out and food cup is formed and it is engulfed. And now the food vacuole is formed. The entire digestion, whether it is after circumvallation or after circumfluence, the entire digestion occurs within the food vacuole. Hence, the process of digestion is intracellular. and the contents of the food vacuole. You try to remember this point. The contents of the food vacuole are first acidic and then basic. Just like in our stomach. For example, in our digestive system, you can see the contents of the stomach are acidic and the contents of intestine are basic. That means in our stomach, HCl is produced, whereas in the intestine, because of the presence of bile, the acidic environment is converted into the basic environment or alkaline environment. Same thing occurs inside the food vacuole, but there are no different parts. Inside the food vacuole itself, the contents are first acidic and they can become alkaline later. This is the logic behind this is in food vacuole, when the acidic environment prevails, what will happen? The prey is killed. And once the prey is killed, the food vacuole contents will turn into the alkaline environment where the prey is digested. That means an acidic environment, prey is killed, and in the basic environment, the prey is digested. That is the logic. And here you can find the fine diagrams of circumvallation and also circumfluence, you can see here. Now, there is one more method called invagination. Here, the glaring difference between other modes of nutrition from invagination is in invagination, toxin is secreted, some poison is secreted so that the movable prey will also become immobile. That means it is a state of stupor, or we can say that it is a state of sleepiness or drowsiness for the prey. That means toxin is secreted, it makes the prey immovable, then it is engulfed. Now, what will happen? You can see the ectoplasm, after contacting the prey, invaginates and encloses the prey in food vacuole. So, in invagination, what is happening? First, the toxin is secreted. This toxin makes the prey immobile, that means immovable. Later, ectoplasm comes in contact with the prey. Then, the invagination takes place and it encloses the prey and it is thrown into the food vacuole. There is one more method called import. This is here you can see the difference. Passive food is drawn into the body, whereas in majority of the methods, it takes active food. Now the passive food is drawn into the body by this method called import. And normally foraminiferans use reticulopodia for ingesting the food in this import method. So in circumfluence and circumvallation, what is happening? The normal pseudopodia are playing a vital role. Whereas in case of this import, what is happening? Passive food is drawn into the body, and foraminiferans use reticulopodia for ingesting the food in this mode. And here you can see one more protozoan. Its name is Peranima, and here there are specialized rods, pharyngeal rods called trichites. Trichites. Here you can see the trichites, and these trichites are nothing but the pharyngeal rods that are found in a protozoan called Peranima, and this help in food digestion, food ingestion, not digestion, food ingestion. Digestion always takes place in the food vacuole itself. So that is about one more method called import, which involves the passive food intake. And one more thing, up to this, we have seen the strategies of amoeba and some other protozoans like Peranima. And coming to the paramecium, you know that paramecium is a slipper animalcule. It has got some mouth-like structure within the cell itself, which is covered by cilia. As a result, this paramecium exhibits filter feeding mechanism. So we can say that paramecium is a filter feeder where there is gush of water entering into the cell and it is being filtered. Food particles are being filtered. And now after ingestion, it may be of any mode. Ingestion may be of any mode. In case of holozoic mode of nutrition, ingestion may occur either by circumvallation or circumfluence, invagination or import, whatever may be the method, the digestion has to occur. And digestion takes place inside the food vacuole. As a result, we can say that digestion is intracellular. And I already made it very clear that contents of food vacuole are first acidic and then basic. In acidic environment, the prey is killed, and in the basic environment, the prey is digested. And here you can see wonderful paramecium. Here there is one mouth-like structure. This is called cytopharynx. Pharynx that is present inside the cell is called cytopharynx. It functions like a mouth. And here you can also see the cilia. Now the water enters into the body of the paramecium through cytopharynx where cilia are present, and the cilia filter the food particles and hand over the same to the food vacuum. As a result, we can say that because these are filtering the food particles, we can say that paramecium exhibits filter feeding mechanism. Once the food is taken inside the food vacuole, here you can see the food vacuole goes on rotating. That means it is revolving inside the cytoplasm. It is exhibiting some movements, some roller coaster movements. It is going, here you can see it is just rolling within the cytoplasm itself. That means this rotation of the food vacuoles, this revolving movements of the food vacuoles is called cyclosis. This happens because the digested contents should be distributed uniformly throughout the cytoplasm. For example, if food vacuole is static and if the digestion occurs here itself, the nutrients will be supplied only to this area. But in order to facilitate uniform distribution of the nutrients, the food vacuoles exhibit rotatory movements, roller coaster movement, and this is called cyclosis. This cyclosis facilitates the equal and homogeneous distribution of the digested nutrients throughout the cytoplasm. And the rotation of the food vacuoles is called cyclosis, of course. And here you can see some examples. Paramecium, this is the filter feeder. And the pseudopodia in case of amoeba are used for capturing the prey in the form of circumfluence or circumvallation, ingestion or import. And here Euglena. Here you can find totally different mode of nutrition. I'll come to that Euglena later. Paramecium is a filter feeder, and here you can see Euglena. Here you can see the diagram. Euglena exhibits holophytic mode of nutrition. Let me explain what is holophytic mode of nutrition. It is a plant-like mode of nutrition. You know that plants have chlorophyll, which traps the radiant energy and converts the same into chemical energy. Euglena, here you can see Euglena is green in color because of the presence of chromatophores, that means chlorophyll. And by using this chlorophyll during daytime, Euglena performs holophytic mode of nutrition. But during night time, chlorophyll is of no use. As a result, during night time, it simply excretes the chlorophyll, it sheds away the chlorophyll, and it switches over to saprozoic mode of life or saprozoic mode of nutrition. So you see the beauty of paradigm shift. Normally we don't exhibit change management, whereas the protozoans, which are called wretched animalcules by Anthony van Leeuwenhoek, they exhibit wonderful paradigm shift in nutrition itself. Euglena during daytime, it is performing holophytic mode of nutrition by using chlorophyll, and during nighttime, because it knows that it's of no use, chlorophyll is of no use, it simply rejects it. It simply excretes the chlorophyll and switch over to the saprozoic mode of nutrition. As a result, because Euglena is performing holophytic and saprozoic mode of nutrition, we can say that Euglena exhibits mixotrophic mode of nutrition. So here you will have the clarity. Mixotrophic mode of nutrition is a nutrition which involves two different methods. So that's about this Euglena. And that's about the process of nutrition, digestion, absorption, assimilation. But what about digestion? What is digestion? Digestion is nothing but the removal of undigested waste material. How it is done? It is done by this cytopise. Cytopise is called cell annus. Annus of the cell is called cytopise. It may be temporary or it may be permanent. In case of paramecium, it is temporary, and it is in case of paramecium, it is visible. That means the cytopise is visible only at the time of defecation. Whereas in case of certain protozoans like Balantidium, the cytopise is permanent. So this is how the food particles will be adjusted. That means undigested food particles will be adjusted. And coming to holophytic mode of nutrition, this is plant mode of nutrition. Chlorophyll is required for this. And I already made it very clear that Euglena exhibits this mode of nutrition. And in one more protozoan, you see this Stentor polymorphus. This contains G.L.N. That means this doesn't contain chlorophyll. Instead of chlorophyll, they contain certain algae called G.L.N. And the association between stentor and Zoochlorella is totally symbiotic. That means for stentor, for this stentor protozoan or stentor polymorphous protozoan, Zoochlorella traps the light energy and performs photosynthesis, synthesizes the food material and shares the same food material with stentor. So that is the symbiotic association between Zoochlorella and the protozoan namely stentor. And saprozoic I have already explained, it absorbs dead and decaying organic matter. And if there are two methods involved in the nutrition, then it is called mixotropic mode of nutrition. And another one is parasitic mode of nutrition that is exhibited by certain protozoans like Entamoeba histolytica, which is present in the colon of human being, and it secretes one enzyme called histolysin, breaks down the mucus and other tissue aspects of the intestine, and then devours it. Sometimes it may cause some sort of inconvenience. You know that this Entamoeba histolytica causes one disorder called amoebiasis or amoebic dysentery. That is about the parasitic mode of nutrition in certain protozoans, of course. And I hope this is the last slide. There are certain food robbers. Normally, there are certain protozoans which act as endocommensals. They rob the food material without causing any disease. They rob the food material that are present in the gut of the human being without causing any inconvenience or without causing any disease. They are non-pathogenic, hence they are called endocommensal. And coprozoic, coprozoic, coprozoic mode of nutrition is also exhibited. Coprozoic mode of nutrition is nothing but depending on the fecal matter, depending on the excreta of human being or depending on the excreta of any other higher vertebrate animal is called coprozoic mode of nutrition. The best example for the protozoan that exhibits coprozoic mode of nutrition is Sarcomonas species. It is nothing but a flagellate protozoan found in the fecus of elk. Fecus are nothing but undigested waste materials. Elk, bison, bear, and other animals. So this is the variety of nutrition that is exhibited by or that is found in the protozoans. Thank you very much, my dear students. Meet you once again.